Angiosperms are the flowering plants which has flower as a special organ for reproduction. Flowers are the objects of aesthetic, ornamental, religious, and cultural value. They are the symbol of love, affection, happiness, grief, etc. To a biologist, it is the site of sexual reproduction. The branch of biology dealing with commercial production of flower is called floriculture. Flower consists of four whorls, namely calyx, corolla, androecium, and gynoecium. The calyx and corolla are not directly involved in sexual reproduction, hence called non-essential whorls, while androecium and gynoecium are directly involved in sexual reproduction, hence called essential whorls. Calyx is made up of sepal, which is meant for protection, while corolla is made up of petals, which is meant for attraction of pollinators. Androecium consists of stamens, which is meant for production of male gametes, and gynoecium consists of one or more carpel or pistil, which is meant for production of female gametes. Some flowers may have nectar glands which produce nectar to attract insects. Structure of male reproductive part. Stamen is the male reproductive part, consists of a long slender stalk called filament and a terminal lobed structure called anther. In most of the angiosperms, the anther consists of two lobes, hence called diathecous. The two lobes are joined together by a narrow strip of tissue called connective. Each anther lobe consists of two pollen sacs. Therefore, a diathecous anther contains four pollen sacs or microsporangia which produce pollen grains or microspores. Transverse section of typical anther shows the following structural features. Microsporangium is circular in outline. Each of the microsporangium consists of four wall layers, namely epidermis, endothecium, middle layer, and tapetum. Epidermis is the outermost layer made up of single-layered parenchyma. Endothecium is inner to the epidermis, made up of single-layered elongated cells with fibrous thickening. These cells are hygroscopic, which help in anther dehiscence. Middle layer is inside the endothecium, which is 3 layers in thickness. Tapetum is the innermost layer which surrounds the pollen sac. The cells of the tapetum possess dense cytoplasm and more than one nucleus. It is meant for nourishment of pollen grain. In young anther, pollen sac consists of compactly arranged homogeneous cells called sporogenous tissue. During microsporogenesis, these sporogenous tissue undergo meiosis to form microspores. Microsporogenesis. It is the process of formation of microspore from a pollen mother cell through meiosis. It involves following steps. The sporogenous tissue present inside the microsporangia or the pollen sac has polygonal shaped, closely packed cells. During microsporogenesis, pollen sac increases in size and the cells of sporogenous tissue become rounded and loosely arranged. They are called spore mother cell or microspore mother cell. This spore mother cell is diploid, which undergo meiosis to form 4 haploid daughter cells which are arranged in cluster. They are called microspore tetrad. As the anther mature, microspore tetrad undergo dehydration and separate from one another. This forms a loose powdery mass of pollen grain. Each microsporangium produces several thousands of microspores or pollen grains. The middle layer and tapetum disappears. So the mature anther wall consists of only epidermis and endothecium. The hygroscopic cells of endothecium helps in the dehiscence of matured anther. In matured dehisced anther, each lobe consists of only 2 pollen sacs. After anther dehiscence, pollen grains are released out from the microsporangium. Structure of pollen grain. The pollen grain is the male gametophyte in angiosperms. The size, shape, and ornamentation of a pollen grain varies from one plant to another. The branch of biology concerned with study of morphology of pollen grain is called palynology. The haploid pollen grain is the first stage in the male gametophytic generation which carry haploid male gametes. Hence, they are very important in the life cycle of angiosperms. Pollen grains also affect the life of human beings. There are many advantages as well as disadvantages of pollen grain. The pollen grains of some plants are highly nutritious and can be used as food supplements in the form of tablets and syrups. Consumption of pollen grain of some plants increases the performance of athletes and race horses. But the pollen grains of some plants causes severe allergic diseases and bronchial diseases in some people. A pollen grain may measure 25 to 50 micrometer in diameter. Its wall is made up of 2 layers, namely the outer exine and the inner intine. The exine is made up of highly resistant biochemical called sporopollenin. It is the protective layer and resistant to heat, strong acids and bases, enzymes, etc. No enzymes are discovered so far which can degrade exine layer. So, the pollen grains can be well preserved in fossil. Sporopollenin deposition is absent in some places that forms prominent apertures called germ pores, through which pollen tube come out during pollen germination. The exine may consist of spines, ridges, and other fascinating outgrowths on its surface. The intine is a thin, continuous layer made up of cellulose and pectin, collectively called pectocellulosic substance. It consists of plates of enzymatic proteins nearer to the germ pores. Inner to the intine layer, there is plasma membrane which covers the cytoplasm. The cytoplasm of pollen grain contains a haploid nucleus. This nucleus undergoes mitosis to produce one haploid generative cell and one haploid vegetative cell, also called tube cell. Therefore, a matured pollen grain is two-celled. The vegetative cell is larger in size and has abundant reserve food materials and large irregular nucleus without cell wall, while the generative cell is small and present in the cytoplasm of the vegetative cell. It is spindle-shaped with dense cytoplasm and a nucleus. In most of the angiosperms, that is, about 60%, pollination occurs at this stage. In some angiosperms, that is about 40%, the generative cell undergo another mitotic division to form two male gametes and form three-celled stage. Pollination occurs at three-celled stage in such plants. Pollen viability. Ability of the pollen grain to perform its function of delivering male gamete to the embryo sac by producing germ tube is called pollen viability. The viability of pollen grain differs from one plant to another and also depends on temperature and humidity. In some plants like rice, wheat, etc., pollen grain may remain viable for about 30 minutes after their release from the anther. In some other plants like rose, brinjal, tomato, bean, etc., it may remain viable up to a month. One can maintain the pollen grains in viable condition by a technique called cryopreservation, in which pollen grains are stored in liquid nitrogen at minus 196 degrees Celsius. It can be used in crop breeding programs. Structure of female reproductive part. Carpel or the pistil is the female reproductive part, consists of a basal bulged structure called ovary, a long tube called style, and a upper receptive part called stigma. The carpel may be one or more than one and may be free called apocarpus or fused called syncarpus. Inside the ovary there is a cavity called locule and a ridge of tissue inside the cavity is called placenta. Ovules or Megasporangia arises from the placenta. The number of ovules varies from one plant to another. The typical ovule or microsporangium shows the following structure. The ovule is attached to the placenta with the help of a small stalk called funicle. The main body of the ovule is called nucellus. It is made up of parenchyma tissue. The nucellus fuses with funicle in the region called hilum. The nucellus is covered by a protective covering layer called integument. There may be only one integument called unitegmic or two integuments called bitegmic condition. At one end, the integument covers the nucellus completely called chalaza, and at the opposite end, the integument leaves a small gap or opening called micropyle. Each cells of nucellus have abundant reserve food material. Among them, one cell undergo meiosis to produce a haploid female gametophyte or embryo sac. Types of ovules. Based upon structural variation, angiosperms may have different types of ovules. Orthotropous ovule. Body of the ovule is in straight line with the chalaza, micropyle and funicle. Anatropous ovule. The ovule becomes completely inverted so that the micropyle lies close to the funicle. Hemianatropous ovule. The body of the ovule is at right angle to the funicle. Campylotropous ovule. The body of the ovule is bent and the alignment between the chalaza and micropyle is lost. Amphitropous ovule. The body of the ovule is curved so that embryo sac and the ovule itself takes horseshoe-shaped structure. Circinotropous ovule. The funicle is long so that it creates a full circle around the ovule. Megasporogenesis. It is the process of formation of megaspore from megaspore mother cell through meiosis. It involves following steps. At the beginning, any one hypodermal cell in the nucellus at the micropylar end becomes more distinct by acquiring dense cytoplasm and large nucleus. This cell is called archisporium. This archisporial cell divides periclinally, forming an outer parietal cell and inner sporogenous cell. The sporogenous cell functions as megaspore mother cell, which undergo meiosis to form 4 haploid megaspores, which are arranged in linear tetrad. Among the 4 megaspores, those cells which are present towards the micropylar end degenerates and only one which is present at the chalazal end remains functional. These megaspore enlarge in its size and gives rise to the female gametophyte or embryo sac. The haploid nucleus of embryo sac undergo 3 successive mitotic divisions to form 8 nucleus. The nuclear division is not followed by immediate cell wall formation. After the 8 nucleated stage, cell wall is formed and these 8 nuclei organizes in a definite manner within the embryo sac. 3 haploid nucleus arrange towards the chalazal end called antipodals. 1 haploid cell at the micropylar end forms the egg cell. Surrounding the egg cell, there are 2 haploid nuclei called synergids. Egg cell and synergids together called egg apparatus. The 2 nuclei from both the poles move to the center and fuses to form a diploid cell called central cell or secondary nucleus. The synergids have special cellular thickening at the micropylar end called filiform apparatus. This helps in guiding the pollen tube to enter into the embryo sac. Since the embryo sac is developed from a single megaspore mother cell, it is called monosporic embryo sac. The embryo sac consists of 7 cells and 8 nucleus. Hence, it is called 7-celled, 8-nucleated polygonum type of embryo sac.